Hello, my name is Brandon Schloman. I'm a grad student at the University of Oregon working with Raghu Parthasarathy. And in this special virtual presentation that should have been at APS March meeting in Denver, I'm going to tell you about my work looking at interactions between immune cells, shown here as these colored blobs moving around, and gut bacteria. Specifically, I'm going to talk about how we can directly visualize these interactions in a model organism called the zebrafish, and how we can control these interactions by manipulating bacterial behavior. Okay, so in general, we know that the immune system has to balance its ability to respond to threats with a certain amount of tolerance to avoid self-damage. But this issue of tolerance is especially important in the intestine, because in the intestine, in addition to the occasional pathogen coming through, there are trillions of resident bacteria always present, making up this thing that we call the gut microbiome. There are many different types of these bacteria, and their activities can be directly sensed by innate immune cells, such as macrophages and dendritic cells, which I'm showing here in green, that are situated adjacent to this epithelial boundary layer, which I'm showing here in blue. So I became interested recently in this problem of how tolerance is achieved, specifically in the gut, how it's regulated, and how it breaks down during diseases like inflammatory bowel disease. One process that we know is important for maintaining tolerance is the simple act of spatially confining bacteria away from this epithelial boundary, for example, by these protective mucus layers. This effectively dampens down the pro-inflammatory signals coming off these bacteria. So today, I'm going to tell you about a bacterial mechanism that we've been studying for how this confinement can break down. This mechanism involves bacteria using swimming motility to effectively escape this confinement and access the epithelial boundary layer and then cause inflammation. And everything I'm going to tell you about today is written up in a paper that is recently accepted for publication in PLOS Biology, should be out soon, and is also on bioRxiv. Okay, so in order to study these intrinsically spatially structured interactions between bacteria and immune cells in the gut, I take a live imaging approach using a model organism called zebrafish. Shown here is a picture of a larval zebrafish. They're quite small, just a couple millimeters long, but their key feature is that they are optically transparent. So this allows us to do live imaging studies where we can go in and directly observe immune cells and bacteria within the intestines of live animals. In addition, there's an increasing number of genetic tools available to this model organism. So this allows us to do things like get zebrafish that have fluorescent markers of various immune cells and reporters of gene expression. And it's important to note that zebrafish, being vertebrates, have immune systems that look very similar to the human immune system, with all the major cell types conserved in both the innate and adaptive compartments. But there's an interesting developmental separation. So larval zebrafish at the age that I study only have an innate immune system. But this is actually nice because it allows us to study this first process of the immune system sensing gut bacteria in the absence of additional complications involving the adaptive immune system. On the bacteria side, we make extensive use of these protocols that have been established for deriving larval zebrafish initially devoid of any microbes or germ-free. What this allows us to do is to start with a clean slate of an empty gut, and then add back in single species of bacteria at a time, and then study how these controlled single species communities interact with the immune system. All the bacteria that we study in these experiments are native to the zebrafish gut. They've been isolated and engineered with nice fluorescent tags so we can do fluorescence microscopy. In order to do fast three-dimensional imaging of these fairly large volumes while minimizing photo damage, I used a technique called light sheet fluorescence microscopy, which is well suited for this. Okay, zooming in on this anterior region of the gut, I'll show you what this all looks like when we put it together. So here's a real-time movie of some bacteria in pink swimming around the intestine of a live larval zebrafish. This fish has been engineered with a green fluorescent protein reporter of TNF-alpha, which is a canonical pro-inflammatory cytokine. So the idea is that these green cells you see lining the gut, just here, here, and here, are engaging in an inflammatory response. Now, these bacteria are a species of Vibrio that is native to the zebrafish gut. It is a common member of the conventional zebrafish microbiome and under conventional circumstances does not cause disease. We see here, though, that these bacteria are capable of eliciting a immune response as measured by this TNF-alpha reporter. We also note that these bacteria, being able to swim, can make frequent contact with the epithelium surface, and we hypothesized that because of this, swimming motility was important in determining this bacteria's ability to elicit immune response, and went on to test this. But before I tell you about that, I want to also show you that we can do three-dimensional imaging of these systems by the same technique of light sheet microscopy. And when we do this, we find that there is really a large number of TNF-alpha positive cells around the gut. And in separate experiments that I unfortunately don't have time to tell you about, involving double transgenics, we discovered that the majority of these cells around the gut that are TNF positive are macrophages, which are members of the innate immune system and are well known to directly sense bacteria. Okay, so in order to test the role of motility in Vibrio's ability to induce an immune response, we took a genetic approach and generated non-motile mutants that cannot swim. So these cells still make flagella, they just can't spin them because of a stator deletion. So when we colonize fish with these motility deficient mutants, we find that they don't swim, but in addition, they become sequestered away from the epithelial boundary and become confined in these multicellular clumps or aggregates, which are likely covered in intestinal mucus. As a result of this, the whole population makes less contact with the epithelium surface, and there are also fewer cells in total. When we go to measure the immune response to these non-motile mutants, we find that the immune response is substantially reduced. So to quantify this, we do computational image analysis and segment these bright green regions corresponding to the TNF-alpha expression. And then what we do is normalize these total fluorescence intensity by the median value that we find in germ-free fish, or fish without bacteria. When we do this across many different fish, each one of these markers here corresponds to a different fish, we find that the non-motile mutants induce an immune response that looks the same as having no bacteria at all, whereas the wild-type Vibrio elicits an immune response that is, on average, 100 times higher than these levels. All right, so this indicates that motility is required for Vibrio's strong immune response in this context. We then wanted to go further and ask whether or not the immune system can sense changes in the swimming behavior of these resident bacterial populations. So to do this, my amazing colleagues at Oregon engineered an inducible switch version of this mutation. Operationally, the way this works is that we can start out with bacteria that cannot swim and colonize the gut in a non-motile state. And then simply by adding small amounts of this inducing molecule to the system, we can turn on the expression of motility genes in vivo. How this works at the molecular level is not important for this talk, but for people who think about this sort of thing, it works by a double repressor type circuit using TET repressor circuitry. Okay, so this is what this looks like. We can start out and colonize fish with these bacteria with a switch in the off state, so they cannot swim. And consequently, they form these sort of a long stringy aggregate localized to the middle of the gut. And then we can follow these population over time after we induce motility. And when we do this, we see the emergence of many planktonic cells that go on to repopulate the anterior region of the gut over here, and then can confirm through live movies that these cells are in fact swimming, right? So with these switches, we can control the swimming behavior of these bacterial populations in vivo. We then follow the immune response to these bacteria over time as they gain back the ability to swim. So when we do this, we start out with populations with a switch in the off state, so they cannot swim and form sort of small clumps, similar to the mutant. And we also see a low immune response. 24 hours after induction, we see the emergence of a subpopulation of more planktonic cells that we verified to be motile, and a corresponding increase in the number of TNF-alpha positive cells around the gut. By 48 hours after induction, we see the emergence of a large population of swimming cells, similar to wild type, and also an even further increase in the amount of TNF-alpha expression we measure around the gut. When we quantify these effects in the same way that we did before, we see this nice increase in reporter activity that approaches wild type levels by 48 hours post-induction. Right? And again, all we're doing here is giving these bacteria the ability to swim. Right? So what these experiments tell us is that the immune system seems to have this ability to dynamically sense the swimming behavior of bacteria and respond to these changes in that behavior really in step with the bacteria. Right? So this then immediately raises a bunch of interesting questions about what exactly are these immune cells sensing, and what sets the time scale of their response, which is precisely what we are pursuing now. With that, I would just like to thank all of the great people that worked with me on this project, in particular Travis Wiles, a postdoc in Karen Gilman's lab, who's a co-first author on the study, thanks to funding. I'll just end by pointing out again that everything I talked about today is written up. It will be out soon in PLOS Biology. It's also available right now on bioRxiv. And if you have any questions or comments, definitely email me. My email is here. And yeah, it'd be great to hear from you. Thanks for tuning in.